You have probably heard it said that what starts here changes the world. And today is about changing the world, that part of it, the second part. And it is our honor and pleasure to have with us today Dr. Robert, MIT Professor Dr. Robert Langer. We hope he's been here before, and we hope he comes again, but he's here today, and we're delighted. My name is Bob Metcalfe, and I'm Faculty Director of the Innovation Center in the Cockrell School of Engineering, where it is our job to encourage faculty and students to start companies, to change the world, to have impact through startups. Now, Dr. Langer, who just recently won the Queen Elizabeth Prize, is a member of all three academies. He can talk about anything for hours on end, but what we're asking him to talk about today is two things. One is, how can you remain a world-class research professor and still have time and energy to start companies to have impact with your innovations? And that'll be in the first session. And then the second session, we're building a new medical school. What advice does Dr. Langer have for us and how to build that medical school so that it's innovative and has impact? In the first section, Professor Nicholas Peppas, who's the head of biomedical engineering, will join us and interviewing Bob. And in the second session, Clay Johnson, Dean of the Dell Medical School, will be here to get that advice.
Good afternoon. Thank you all for coming. Bob, thank you. It's really a great pleasure to see you here. I know you woke up at 3:30 in the morning in Madison, Wisconsin to make it here. I'm delighted you are with us. I want to give a small introduction before we go into pursuing innovation. Bob Langer is like all of us, a classical example of American ingenuity. He was born on August 31st, 1948 in Albany, New York, comes from a family that had a father who was a small businessman. In fact, he was a liquor store owner, and a mother who was staying at home taking care of two kids, Bob as his youngest sister. He got very excited because when he was in primary school, his grandfather was playing mathematical games with him. And then in high school, or junior high, he started getting these Gilbert sets, if some of the old-timers remember them. One on chemistry, he loved it, and he was at home playing with it. He decided to go to chemistry. He went to Cornell University in 1966. And while he was a graduate of the Mills High School that some of you know in Albany. Then after that, he went there, and while he was there the first semester, he loved chemistry, but he decided to pursue chemical engineering, Cornell. There was a professor by the name of Bob Finn that was a biochemical engineer that really helped him in that decision. After he finished, he moved to MIT. He started working mostly in the biochemical field. He finished MIT. We were classmates, of course, at MIT, working in the same floor, proximate laboratories. And there was the big decision. He had many, many, many offers from oil companies. He was a chemical engineer. He still is. He decided to do something different. In 1974, he decided to join a medical group. Professor Judah Folkman at Harvard. He was a young man at that time. He was collecting unusual engineers and individuals like Bob. And he went to that laboratory, and the rest is history. He started working on angiogenesis with Judah Folkman. That's where he met people that have become other legends in the field, like David Kessler, who became an FDA commissioner, like Henry Brem, that probably he will mention in his work, and of course Jay Vacanti, with whom he started the tissue engineering. He started at MIT in 1977, first as a visiting professor, and then in 1978 as a regular assistant professor. Not in biomedical engineering, not in chemical engineering, but in nutrition and food science. Why? Because in those days it was difficult for chemical engineers and even biomedical engineers to accept the idea that an engineer would be working on problems related to the biological field. And he has many stories to tell you about it. In those very early days, he came up with the first patent. It was a patent that was a technology with which he was trying to release, to deliver macromolecules from polymers. And all of you who have heard polymers, you know how difficult that is. Well, Bob was able to do it in an ingenious way. It took him 7 years to have the patent issued. The patent was issued in 1993. And eventually, after a lot of efforts that he may decide to tell us about, it ended up going to the company Alkermes, and it became the Prolise technology. It was one of the first technologies, and then followed many other technologies. In 1989, he became professor of chemical engineering. He's now the institute professor at MIT. There are only 12 institute professors. Bob has really been a tremendous innovator, an entrepreneur, yet at the same time a fundamental scientist. He continues doing fundamental work in a number of subjects, while at the same time carrying out really all the more entrepreneurial activities that he has. He's a tremendous inventor. For those of you who don't know it, he has 1,100 US patents. If you add the international ones, probably many more. He has about 1,300 publications in refereed journals, about 100 of them in Science and Nature, and I don't know, maybe about 40 in the Proceedings of National Academy of Engineering. For those of you who count H indexes, 209, and citations about 167,000 last night. By far, by far the most cited, and actually Google Scholar has forgotten here most cited paper, the paper with Vacanti in Science, which is about 9,000 citations. For some reason, it's not mentioned. So add another 9,000 there. He has been recognized everywhere in the world. I don't want to embarrass him too much, but some of us have honorary doctorates. Bob has 24 honorary doctorates from Harvard, Yale, Louvain, ETH, everywhere in the world. Bob has received many, many awards, not only from the chemical engineering and the biomedical engineering and the control release community, but more importantly, he has received the Wolf Prize from Israel. He has received the Millennium Award, which is the equivalent of the Nobel Prize from Technology. It was given in Finland. He has received several other major awards from the American Chemical Society, like the Perkinel Medal, the John Fridge Medal. Some of these awards have been received before by luminaries like Guglielmo Marconi and Thomas Alva Edison and so on, and that puts him in the same list to understand really where he is. From the United States, he has received both the the National Medal of Sciences and the Medal of Technology and Innovation from two different presidents, President Bush and President Obama. The latest recognition, the Kyoto Prize from Japan last year. The latest recognition, he will get it in October, is the Queen Elizabeth Engineering Prize. I know he doesn't like me to talk about money. It's £1 million. Last year, he received in Hollywood the Breakthrough Prize, $3 million. These are prizes. These are not awards for research. From Michael Hall, believe it or not, in a very, very interesting, I guess what I'm trying to say, this is a man who saves people. This is a man who comes up with new ideas in solving problems, who helps people with his companies, and he is a great entrepreneur. Before I finish my introduction, I'd like to tell you the companies that he has started. Accusphere, AIR, which is advanced inhalation technology research, Arsenal, Bind Therapeutics, Blend Therapeutics, Eco Therapeutics, Enzatec, which is part of Alkermes now, Gecko Biomedical, In Vivo Therapeutics, Kayla, Microchips, Momenta, Pixar Bio, Pervasis, Volmatrix, Selecta Biosciences, Semprus Biosciences, Seventh Sense, SQZ Biotech, Teris, Transform, T2, and two that I left last. Moderna Therapeutics, that all those of you who follow it at this moment is standing on $900 million funds that they have raised for the research. I've never heard any company which is not public having that much money for research. And the last one is one that is dear to our heart of all of us, Living Proof. If you don't know, Living Proof is the company that Jennifer Aniston is advertising with the hair. And there are some nice videos where you can see Bob and Jennifer Aniston together. So we are here, Bob, the other Bob Metcalfe and I, to ask some questions today about how you do it, Bob. How do you do it? How can you have 100 papers in Science and Nature and at the same time have all these companies and run all over the country? And by the way, if you send him a message, not right now, he's going to send you a message back in 3 minutes. That is something very well known. He answers to everybody immediately. Thank you for coming, and I'd like to ask the first question. How is it possible to do all these things together? Welcome.
Well, thank you. Well, thank you, Nick, very, very much, and Bob, and thank you for that really very, very nice introduction. I don't think anybody does introductions better than Professor Peppas. And I've seen it many times. You know, I would say, I mean, you know, now it almost seems kind of natural to do, for me, you know, what happens is, you know, we work in the lab like anybody else does. We have students do theses or postdoctoral projects. But one of the things that over the years has become, I think, increasingly important to me, and something I did not realize when I started my career, is I thought if I just did the work and our students did the work and we published it, well, people would use it. And, you know, that happens some of the time, but very rarely. I've found that if you're not your own champion, very few people use it. And so that really was the initial motivation. I can go into more detail on that for getting these companies launched. And with the companies, and most all the time, it's been with students or postdocs of mine or former students or postdocs of mine. You know, we've done these. And what I could see, some of it goes back to the early 80s, that when we have these companies, they can do things that I could never do otherwise and that our students could never do otherwise, which is basically manufacturing, clinical trials, create products, you know, that get out to the world. And so to me, what I could start to see is that the impact of what we were doing in the lab was really multiplied tremendously by these companies. And I think my students over time would see that, and you know, they've continued to do it. And so it kind of then gets a life of its own. When the current students see what the former students have done, they say, well, this is a career path that you know, seems interesting to me, seems important, seems where I could make an impact. And they do it as well.
But I assume at the beginning it was difficult. I mean, if we go back to the late 70s, and early 80s, I mean, when you went for the first time to the Office of Technology Commercialization, whatever you call it, they looked at you. Did they help you immediately? Is it easy? Was it easy, or?
No, it was actually it was very hard from a number of standpoints, and part of it's not necessarily having the realization of all the things that would be key. I mean, one of the first things that ended up being important was a patent, and we filed a patent. And I remember 5 years in a row, the examiner turned that patent down. And, you know, finally, the lawyer for the hospital told me that we were just wasting a lot of money and wanted to stop working on it. And, you know, and I realized that trying to explain to the examiner scientifically why we should get that patent wasn't working. I mean, I tried that 5 or 6 times, and we kept failing. So I was trying to think, how could we convince the examiner to allow this patent? You know, legally, of course. And one of the things that I ended up doing was I realized that when we first started making some of these findings, a lot of people felt that you couldn't make them, they were impossible. So I actually went back and did what's called a science citation search. I searched our paper, which we had actually written in Nature, and see what other people wrote about it. And I found a quote, actually, from a number of people where they said that the work we did was extremely surprising. It went against conventional wisdom. And I showed that to our lawyer, and he went down to talk to the examiner and showed that quote to the examiner. And the examiner actually said to him, he said, well, I tell you what, I didn't realize that. I'll allow this patent if Dr. Langer can get affidavits from each of the people who wrote that they really wrote it. And so we did. They were nice enough to write back that they really wrote it, and we got that patent. So that was the first thing. But then the second thing is, I'd worked on, but having that patent was important. Second thing that happened is I thought, several large companies contacted me after I'd worked on this stuff for 10 years. I mean, I kept thinking, if we wrote it on stuff, people would just use it, and they weren't. But then after about 10 years, a couple companies contacted me, and they gave me, and I was a consultant to them, and I actually got a significant grant from them. And these were both multi-billion-dollar companies, one in human health and one in animal health. And so they gave us grants, they gave us a consulting fee, and they even were going to do work on it themselves. And I was very excited about this. But what happened is they would maybe do an experiment or two a year, and when those experiments didn't work, they kind of gave up. So a couple years later, one of my friends at MIT, Alex Klebanoff, said to me, he said, Bob, we should start a company ourselves. And I was able to get those patents back. And at that time, you could get, we had several venture capitalists come to our office and they talked to me about possibly wanting to do a company. And so I was able to get those patents back. And we did start a company to make these products. But it took a long time, and it also, and even then when we did it, we ran into problems like our first CEO wasn't very good. I had no idea how to hire a good chief executive officer. And so, there were all kinds of issues. And, you'd keep learning, and mostly by making mistakes.
So 15 years ago, I became a venture capitalist and joined your favorite VC firm, Polaris, to learn venture capital. And Terry Maguire was teaching me how to be a venture capitalist, like due diligence and how you have to check everything. And that first week on the job, Terry invited me to a meeting with you and Ram and Ganesh. And you presented 5 PowerPoints, roughly, describing the field of glycomics and the sequencing of sugars that coat molecules and stuff I don't understand. And after the presentation, Terry Maguire stood up and shook your hand and made the $1 million investment that you were seeking. Of course, after the meeting, I went to Terry, and by the way, I kicked in $100,000 into that deal, and thank you very much. I asked Terry about the due diligence part. We had this meeting, and he gave you the $1 million right there. And I said, Terry, what about the due diligence? And he said he had done his due diligence on you 20 years ago. So he was ready to go. So that company, could you tell us that company which had a phenomenal first year?
Sure. Well, so Momenta, I mean, that's actually a good example of the kinds of things that we do. So I had a couple of a student, Ram Sasaskaran, and then a postdoc, Ganesh Venakatraman, and one of the things we worked on is certain enzymes called heparinases that we had isolated. And then actually Ram, for his thesis with me, actually cloned the genes for it. And then we kept going further and further, and one of the things that came out of some of this work, which is quite complicated, is nobody had ever sequenced what's called a complex polysaccharide before. The heparin, for example, which people may know of, is a polysaccharide. And the reason why it's complicated is that if you ever looked at it like a protein, like insulin, all the molecules in the insulin mix are the same. That's also true for DNA. But for polysaccharide, that's not the case. That's also not the case for glycoprotein. So they're more complicated. I won't go into the science that much, but we came up with a combination of technologies using enzymes and mass spectroscopy and mathematical modeling that enabled you to sequence polysaccharides for the first time. And Ram and Ganesh spent years on this. And they really wanted to see this get out and get useful, and they wanted to start a company. And we had talked about this probably for 6, 7, 8 years. And finally, we did do that, and I talked to Polaris, and I didn't realize that you had put in the $100,000, but I'm glad you did. I think it's probably worth quite a bit more than that now. So at any rate, and then we also got a very good initial CEO, Alan Crane. That's also very important. And Polaris, you're right, just put in $1 million initially. So the first thing that we thought about doing is that we could actually, there was a drug called Lovenox, so it was a $4 billion a year drug, and that Sanofi made. And it's interesting, we actually talked to them about doing some type of arrangement, but they never thought we could sequence it. But we did, and what happened is that led to making what's called a biogeneric. And the FDA actually allowed the approval of this. Simply, it's complicated, but if you had a small molecule like aspirin, you can show what's called equivalence simply by doing, a chemical identity test, something like NMR or something like that. With a complex molecule, it's very hard to do. But we could do it. We could show chemical identity with the techniques we had. So the FDA gave us an approval in 2010 for this Lovenox, even though it turned out, many much larger companies have been trying to do it much longer than we did. So what that led to actually was the largest syringe launch in history. In the first year, the Momenta product made over $1 billion, and the company has done quite well. It's now working on others as well. The other thing, though, that I always am very proud of with Momenta is that also happened, people may remember this a little bit, was that 7 years ago in 2008, there was this Chinese heparin crisis. It turns out that heparin came in from China, and in 22 different countries, people died because it turned out the heparin was contaminated. I also had been chair of the FDA's science board, their highest advisory board, so I knew quite a few people at the FDA, and they contacted me and they contacted Ram, and they said, could we help them? So what we did is we worked with Momenta, and actually the FDA and the CDC, the Center for Disease Control, to look at this contaminated heparin and understand what made it contaminated. And we identified the contaminant. In fact, there were 3 papers that we published with the FDA and the CDC, 1 in Nature and 2 in the New England Journal of Medicine. And so we identified the contaminant, established new standards by which heparin could no longer cause these deaths. And after that, the deaths went to 0, essentially. So that would never have happened without these sequencing techniques that the company had. So, so that was another kind of benefit that came out of all this.
But you were, in that transaction, catalytic. You're a professor at MIT. You present to the VCs. Ram and Ganesh go off to start the company. Terry brings in Alan Crane. And meanwhile, you're back at MIT doing your next research project, right?
Right. Well, I've always done, in other words, all these things, I should say almost all the companies have started because they had their origin, say, in our lab or with a collaborator. And we're always doing the next project. I mean, a lot of these projects are going on simultaneously. But, we've been working on a number of things for many, many years. But sometimes something reaches a point of maturity and where it really makes sense to have a company because you're at that point where you could move sort of from research to development to create this product. And secondly, we may have people involved who have done this, like, say, Ganesh, who, maybe they've been in academics for 10 years now. They really want to work at a company because they feel this passion to take their discovery and get it out to help improve people's lives.
Was it hard to convince Ganesh that he should start Momenta?
No, actually, quite the opposite. I mean, he had wanted to do it for a long time. But, raising money and being far enough along, that's never trivial. But he had seen me do it before, and I think he really wanted to do that. Today, he supervises about over 300 people, and I think it's been very rewarding for him in many ways to have had this opportunity. But he was interested in it. But lots of times people are interested in it, but the difficulty is, in fact, sometimes people ask me, when is the right time to start a company? And a lot of times people sometimes start too early, sometimes they start too late. But I mean, what I usually like to have is good intellectual property. I like to have at least some good papers that we've written in top journals. I like to have, especially if it's in the medical area, some type of in vivo proof of principle that it really works. I think if you start too soon, especially in medicine, investors get tired. Of course, if you start too late, people will beat you. Or it'll never happen, obviously. So you have to figure out that sort of right time to do it. But to me, the right time meets those criteria that I mentioned.
Along those lines, it's very important for us. Quite a few people in the audience are graduate students, postdocs, professors who have IP. At what point do you make the decision that it's time? I mean, it's obvious. You start working on the project. I remember the old days, you or your students would file for a patent very early, very early. What is your advice? At what point do the faculty or the students, do they go and file? And what do they file? A disclosure, a provisional? Do you file provisionals or do you go directly for the patents? And what about animal studies? More and more these days, a company will not talk to you. For the medically oriented companies, they will not talk to you unless you have some animal studies.
No, I agree with all those points. I mean, what I would say is, I certainly want to file a patent before the paper's in, but I would never hold up the paper for the patent. For example, we might do, I'll make just an estimate. If we wrote a paper, we might do 10 or 15 drafts of a paper. I'd say by the time you're at draft 2 or 3, that draft is probably good enough for the patent. In other words, it doesn't need to be totally finished, because the patent is more on the concept and the idea, rather than fine-tuning, making sure all the statistics are 100% clean and so forth. So I think that you can use the paper as a basis for the patent. You certainly don't want to file a patent after the paper, because then you lose it. It's going to be prior art. So I'd say that an early draft of a paper, to me, is a good time to file a provisional patent. I mean, you could file it earlier, but generally that's what we've done is what we've said. And you're right, Nick. The animal data is very important. That animal data may or may not be in the patent, because it's a question of what you're putting in the paper. But if you want to get a company or investors to like the idea and put money in, they will want to see some in vivo proof of principle animal data, and that may come later. I mean, you probably will need it, and you probably will want it anyhow. Even if you could get the money, the problem is you'll end up getting diluted tremendously if you start too early too.
And at what point do you approach a VC? I know you work with Terry McGuire quite a lot, with Polaris, but I'm pretty sure you have other companies. I mean, when do you know it's time for you to approach investors?
Well, I would say when we have, if what I'd like to do, if it's possible, is to go in with a bang, have a really powerful paper, Nature, Science, some journal like that, know that we filed the patent, have really good data, go in right before that paper's going to be out.
Okay.
So you've been seen hanging around with Jennifer Aniston.
Yeah.
And I remember the day that Amir Nashat came to the partners meeting, and his jet black hair was shiny and beautiful. And somehow I linked those two events. Were they related?
Yeah, I think they were. So anyhow, this is the story of living proof, which Nick mentioned. So, and Amir Nashat was one of my graduate students. He got his PhD with me and ended up choosing a career in venture capital. He's been incredibly successful as a general managing partner at Polaris now, which manages about $3 or $4 billion in funds. But the story of living proof, I mean, most of the things we've done, I mean, most of my research it's been kind of basic kind of engineering and science, but if anything, applied to medicine. But one day, and we've started, most of the companies have been applied to medicine, but one day, Amir and John Flint, who is another Polaris partner, came to see me and they said, well, you've done all these companies in medicine, could you do anything in hair? And so it's interesting, we took a look at that, and and the science actually in the hair area isn't real good. The first problem that they brought to our attention, they said, well, they had all these hairdressers. I said, well, is frizzy hair. They said, could you ever come up with something that could treat frizzy hair better? So we actually looked at that, and it's amazing. There's about 50 products on the market today for frizzy hair. Every one has the same active ingredient, which is silicone. And basically the idea is you want something that what we call is hydrophobic to keep water out. What causes frizz is moisture, humidity, and things like that. So we actually just took a much more fundamental scientific look at this and said, well, are there materials that are going to be even more hydrophobic than silicone? We found actually an FDA-approved material, a polyfluoroester, that keeps moisture out much, much better than silicone. It's actually much lighter, and that became no frizz. Second thing they asked us to do was, and that may have been what Amir was wearing, but second thing they asked us to do is they said, well, can you come up with ways of giving hair more body? And so actually Dan Anderson, who was one of my postdocs, he's now a professor at MIT, one of the things we did, and again, it was fundamental chemistry, is we created these libraries of polymers. And the whole goal was initially was to do this for gene therapy or delivering siRNA. But since we had these libraries, you could really use them for anything. And we file patents, right? And the patents are composition of matter. So meaning that you could use them for anything. In other words, you could use them for gene therapy, you could use them for hair, you could use them for anything. So we actually did some screening. We found one of those polymers, which is a polybeta amino ester, created an incredible amount more body. And that became full. Actually, now Living Proof has like over 50 products based on these. And Jennifer Aniston's actually very involved in it. She's a significant shareholder and spokesperson. If you drive by the building we have in Cambridge, her pictures all over it. Probably looks a little better than mine would. And so it's been quite exciting. So I think it's leading, Sephora's leading set of products. And I have to admit, before this, I didn't know what Sephora stores were, but my wife and daughter did. So it's been an interesting application of science to hair care.
So that's how you pronounce it, Sephora?
I think we have people here who could probably do a better job than I, but those are a class of stores that just sell different kinds of.
Now, you don't own Sephora, correct?
I know. I mean,
but you don't. It's not yours.
No, Sephora's not. Living Proof is ours, but Sephora is, yeah.
So Amir said that at Living Proof, the criteria was they would make a product if you could see the difference across the room.
Yeah.
And then at Polaris, we could tell when a new product was coming from Living Proof because Amir would show up and he would walk in and we could see the difference across the room.
It's kind of different than doing something under a microscope. Yeah, I mean, to do it like that. You're right.
You have in the audience people that belong to a circle of inventors, entrepreneurs that meet very often in Austin, of course, under the leadership of Bob Metcalfe. And the question is, for one thing, have you ever invested yourself in your own idea? And second is, what type of risk do you usually take as an inventor and as an entrepreneur yourself? How do you know when to stop?
Yeah. So first, have I invested? And the answer is yes, but I haven't invested that much. In fact, it's funny. Usually, people think if I invest, then I must think this is great, and really they should invest too. But actually, really for me, almost the opposite's been true. Sometimes the companies have had difficulty. In other words, they're not all they don't all go like a straight shot and do wonderful. Sometimes they have difficulty, and I realize like if I do invest, other people might invest, and it just gives the company a boost. And I've done it for that reason more than any other reason. It's not like I invest enormous amounts of money. I mean, I certainly don't have I don't manage $4 billion yet, but like Polaris. But I do invest, and I do think that it's made a difference in terms of not just the money, but really the support and other people investing. And then the second part of your question, Nick?
When do you know when to
When to
stop?
stop.
And when it's getting to...
So I don't stop that quickly. I mean, I've never been very good at stopping, but of course it's a team effort. If it's a company, what happens is it's a decision of everybody on that. But most of the time when we've done these companies, they've been based on what I'll call sort of a core technology, like microspheres that could deliver a drug. And of course there's 100 drugs. And what's going to turn out is it's going to work well for some and it may not work as well for others. And that's really been true of almost all these companies, which is part of, I think, what gives them a chance to succeed, particularly if we're smart enough to pick the right ones first, which we often aren't. But if I believe in the science and I feel that the technology, there's enough of a market, I know some of them will work. And in pharmaceuticals, for example, you could have 9 failures and 1 success, and investors are going to do extremely well if that occurs. because some of those are going to be worth billions of dollars.
Have you had failures?
Well, we certainly have had failures.
Did you start companies that didn't go anywhere? Or?
Actually, I'd probably say not yet, but that's certainly possible. But we've had failures, like with the microspheres, for example. I would say the first 8 or 9 different drugs we put in the microspheres never led to products. Some were technical issues, some were partners dropping out, some were manufacturing issues, but eventually we found some that ended up being very successful and used by millions of people. But so there were lots of failures, partial failures.
I have been asked by colleagues, and especially my dear friend George Georgiou in the audience, to ask about Moderna, and I would like to ask about Moderna. But before we go to Moderna, I want to ask about another company in your early days, AIR, Advanced Inhalation Research.
Sure.
And the reason I'm asking is because I recognize in the audience there are quite a lot of colleagues who love to do fundamental research. That's what they like to do. And you remember, we come from a generation where we learned that as an engineer, as a scientist, you do fundamental research. Can you tell us a little bit how this fundamental work really helped in developing what finally advanced inhalation research did?
Sure. So I had a postdoc in the 90s named David Edwards. And actually, when David came to our lab, he had actually never done an experiment. But I had talked to him about this aerosol area as a means of drug delivery. And for reference, aerosolization is an unbelievably inefficient process. You're lucky if you take an aerosol and 2% goes from the device into the lung. And what that means is that there might be lots of drugs that you might want to deliver that way by an aerosol, but you won't, because 98% that you're losing, it could be incredibly expensive. It could also mean that you're going to have a giant canister to carry around because you only are able to deliver so much. So just to take a step back and go over why you only get 2%. So people always thought before we got involved that aerosols had to be tiny, really, really small, like 2 microns. And the reason they thought that is that they thought that from studies they'd done, if they were any bigger than 2 microns, they'd settle in the back of the throat. But the thing is when they're 2 microns and you put them in an aerosolizer, it's like wet sand in a way, right? The surface area is enormous, so the aerosols stick together in the device, they stick together in the air, they stick together in the mouth. By the time you get to the lung, you don't have very much. How did people try to solve that? Well, they tried to solve it by mechanical engineering approaches, designing better aerosolizers that would break apart those sand-like aggregates. And maybe if they were really lucky, they got from 2% to 2.5% or 3%, which is actually not bad, right? You could save 50% of the cost, make your device 50% smaller. But interestingly, nobody, before we got involved, ever thought about changing the aerosol. And you might think, well, how could you do that? Like, if you think about aerosols, most of them were liquids like water, like metered dose inhalers or nebulizers. A couple people had made dry powders. But the interesting thing is that whenever you looked at the density of those aerosols, it was pretty much the same as the density of water, like what's called 1 gram per cc. So what David Edwards started doing was modeling studies. And we came up with the idea is, what if we made aerosols that were really big, but incredibly light and porous? And when he modeled that, he found that these aerosols that were really big, like 10 or 20 microns, but very light and porous, did not settle in the back of the throat. Now they floated aerodynamically into the lung. But because they were big, they didn't stick together like the small ones, right? Wet basketballs aren't going to stick together like wet sand. So we were able to get much more in, actually by a factor of 10 or 20. Also, because they were big, they lasted a lot longer. There's a process called that happens normally. There are cells in your lung called macrophages, and they eat particles. So before, what happened is they were like eating a small meal, like a little aerosol. Now, they kind of ate a big meal, like this bigger aerosol. Just to give you a sports analogy, so if you look at what aerosols looked like under the microscope before we got involved, they looked like little golf balls or little baseballs, right? Density of 1. Ours just look like wiffle balls. It's really that simple. By changing the geometry, we changed everything. We could get maybe 100 times more in eventually and make it last a lot longer. We published that in Science. We got very, very broad patent. And that really fundamentally changed how aerosols are delivered. Polaris invested it in it, and I think their IRR, which is the annualized return, that was over 1,000%, which I think is the highest in history. I mean, every company in the world wanted to, Pfizer, Lilly, Shearing Plow, they all wanted to work with the company, and then people bought it, and David didn't want to, run it forever.
And that started from a truly fundamental science from somebody who had never done an experiment.
That's correct. Yeah, and David's had an enormously successful career. I mean, both academically, he's a chair professor at Harvard. He's a member of the National Academy of Engineering. He won a Professional Progress Award, many other awards, and totally started from fundamental research.
Yeah.
And he owns a restaurant in Kendall Square called Art Science.
That's right.
And you can go and inhale your food there.
Well, that's right. Well, he also,
That's serious.
So one of the things that's happened is when people do some of these companies, they get a taste and they've enjoyed it. So he started 3 or 4 companies, all actually in the aerosol area. He had a second one called Palmatrix. He has another one where he's got aerosolized, like coffee and aerosolized chocolate. And then he even created a restaurant, which is getting fantastic reviews. It has aerosolized wine, but it's got very good food, too. But he's a real entrepreneur in every way. And like I say, incredibly successful professor at Harvard. But he's also made quite by the way, the other thing that happened when he did this, he made quite a bit of money. He bought a building across the street from the Boston Public Library and created this foundation called the Cloud Foundation to help poor kids in Boston with arts and science and dance and things like that. So, I mean, it's just a great human interest story. It's wonderful for me to see my students and postdocs, do so well.
Before we get to the big drug company, I wanted to ask how you measure success. A lot of startups brag about how much money they've raised, how many people they've hired, how big their revenue is, and I've often heard, connected with you, a different metric. 2.5 billion, I think, is the...
Well, the metric is helping people. I mean, to me, the reason we did it, and I think that's true for lots of entrepreneurs. I mean, you don't do it, and I think this is, and you talk to people like Bill Gates or others, I don't think that people do it to necessarily make money or even to make money at all. People feel a passion that they want to change the world, they want to do something important. To me, the metric is, are you helping people? Are you improving lives? Some cases, are you saving lives? And all these examples we're talking about, like Momenta with the new heparins and Air with new aerosols that are leading to new treatments for Parkinson's and maybe diabetes and others. I mean, to me, the metric is helping people. But I think if you do that and you make an impact, everything else flows with it.
Talking about the impact, I know that many of you in the audience have followed what Bob has done. Not everything is companies, and really you get very emotional when you look at some of his slides and you see a child, 1 year old, with a big belly because of failure of liver, and how he and Jay Vacanti and others have been able to save such kids. Or when you realize that, probably correct me if I'm wrong, the first brain tumor treatment using BCNU in patients that were about to die 2 days later was done by Henry Brehm and Bob. I mean, these were not companies, am I right? Eventually, yeah, there was a company, Nova, and somebody is selling something now, but there the success is more helping people and really coming up with important solutions. The same thing with the baby that you had who didn't have a sternum, and you were able to recreate a sternum because can you imagine you have a 5-year-old boy that cannot play baseball because the first time the ball hits him, he's going to die, and you were able to recreate, so tissue engineering and so on. Right. So not everything is companies, am I right?
No, that's true. I mean, sometimes I would break it into 2 categories. I mean, sometimes you can do things short of companies. In some of those cases are physician-sponsored INDs. But what the companies enable us to do is if you want to affect a broad number of people and you have to have a manufacturing plant that is going to make the same product over and over and ultimately get FDA approval, the companies end up being critical. So sometimes we can help specific individuals with specific problems under physician-sponsored INDs. But if we want to really affect the masses, then I think the companies are critical because you need to, A, have the manufacturing capability, and B, the clinical trials.
So I'm coming to my last question, and then if Bob Metcalfe allows it, we will open it for a few questions from the audience. And that's Moderna Therapeutics. Now, what I want to ask, and of course, George, you can help me, is here's a company that started with you and several others, Afian and so on, and you started going out and getting funds for this company to the point that you got about 400 million the first time. The company did not go public. You were not talking too much about what the company was doing. So we all knew that this was a very major discovery. And then it was announced, what was it, 2 months ago? You got another 450. So you are sitting on a total of about 900, sitting quote unquote, $900 million, which is to me unusual. I mean, I've never seen anything like this. How did you do it? As much as you can say what's happening in a company
Sure.
like this, and what is the exit plan, for a company like this?
So the idea here, one of my colleagues actually at Harvard, Derek Rossi, had made a discovery that you could take a certain type of RNA, and people had tried this before, but if you give the RNA and you want to try to do some type of genetic therapy, usually you get what's called an interferon response. People get sick. He had come up with a way to do that on stem cells, to create stem-like cells where that didn't happen. And so he came over to see me one day and asked, could we start some better stem cell company? And I looked at it and I said, well, we could, but I said, we can do a lot better than that. It just seemed to me, why should we limit it to stem cells? You could really use this as a new way of producing therapeutics like erythropoietin, or you could make vaccines, or you could do almost anything. And right now, if you try to do a lot of those things, you do it with proteins. So that might take a year to make, and there's all kinds of limitations. Here, you could just give a little bit of this modified RNA, and the body will do everything else for you. So that was sort of the idea. And then the next day after he had talked to me, he actually talked to a venture capital group, which in this case it was Flagship, who we'd also done work with, and they were excited about it and wanted to put some money in. Initially, we actually didn't raise that much. We probably raised about $1 million and mostly filed patents. And it was something we kind of kept in stealth mode because we didn't really want to tell everybody we were doing this. We wanted to file, do the experiments, get the intellectual property. When we had done that, and we also had then gotten a very good CEO, Stefan Bonsell, then we raised some more money at a higher value, and we've kept doing that. So now the goal really is to have it in the clinic, maybe with about nine different diseases, within 2 years, and many more after that. And we hope, you never know, we hope it'll become the most successful biotech company ever, and I think it is a revolutionary technology. I think the exit, I don't see it. To us, we could go public, could get bought. I think we'll go public at some point when we really want to raise another giant chunk of money, perhaps, though we raised, like you said, a pretty good chunk of money without going public, and I think we could do that if we wanted as well. So I think the exit is just to make the greatest company in the world to be a public company.
Bob, you used the word we a few times there. What is your role at this company?
At this company and almost all of them, I'm a founder and I'm a member of the board of directors, a member of the scientific advisory board. I never left MIT, but Moderna is across the street from me, as are a number of these. That's one of the nice things that Kendall Square, has become now the largest concentration of biotech companies anywhere in the world. And so, when I want to go to Moderna, it's less than half a block from my office. Living Proof is about 2 blocks. A number of them are all very close, so I can just, take a walk, or they can come see me. But most of the time, my role has been board member, founder, and the kinds of things that I help with are, trying to help recruit the top people. Sometimes it's raising money, and sometimes it's troubleshooting on the science. Mm-hmm. But whatever it takes to get those products out there and to make that company great.
We have some time for some questions. If anybody in the audience would like, yes, please.
Dr. Langer, my name is Harvey Fry. Two-part question, please. In your business model, taking ideas and science to the marketplace, what are the one or two things that tend to frustrate
Sure. Well, I think sometimes what's frustrating and unpredictable are issues with regulatory authorities that you just, this is in the medical area, that you just can't predict. I mean, another thing that's frustrating sometimes is usually when you start these small companies, you're working with large companies, and sometimes there are people who are at those companies and they are champions, but then they maybe leave or move or something. How can you prevent it? Really, the only major way to prevent a lot of these things is with a lot of money, because you don't, I mean, you're always going to get failures. It's very, very hard to know when you're going to, when. Well, that's right. It buys you more time and gives you more shots on goal. So when I look at some of the companies that have been extremely successful today, they've made a lot of mistakes, but they've had enough money that they can weather that storm. So your point about, yeah, buying time is right, but it also buys you more options, right? In other words, lots of these companies, like I was saying, are platform technologies that you're applying across the board. If you only have enough money to do the one and it doesn't work out, you know, you're over. But if you have enough money to do 5 or 10 and one works, like I was saying earlier, that one will be worth another 20 that don't work. Yes. However you call.
You used a key word that I reacted to, champion.
Yes.
And how did you establish that for yourself, and how could we parametrically understand it as an audience, what it meant to you, and how it has helped you get where you have obviously gotten?
Yeah. Well, so for me, I mean, it's just, and let me answer this in a couple ways. For me, I just believe in what I do, and I wanted to not just, like I say, write papers. I wanted to get those ideas out to the world, and so I guess I would do whatever I could to make that happen. My students would see that, and maybe in that sense I was a role model. So some of them, that's a career path that they have wanted to choose also. The example I often pick when I talk about champions actually isn't me at all or our lab. It's actually Steve Jobs and Apple. I think it's just a wonderful example of champions, and it'll illustrate a point or two that will get to the last point you raised. So what I found, so Steve Jobs, as people may know, he and Steve Wozniak, they were these two young guys. They started Apple Computer, got off to a pretty good start. But then the board thought that, you know, they didn't have enough experience. They wanted to get a professional CEO, so they brought in the guy who was president of Pepsi, and they fired Steve Jobs. Over a 15-year period or something like that, they brought in 5 more professional CEOs. None of them, each drove the stock down further, until they finally made a merger and got Steve Jobs back, and then it became the most valuable company, still is, in the world, because he was a champion, in my opinion. And I think the lesson to me in all these things, and it's never simple, but I always would trade passion over experience. You know, a lot of times investors will feel they really want to have this person who's got enormous experience, and experience is useful. But the willingness to walk through walls to make your technology work, to me, that's more important. And I think having that idea in mind when you do these things, you know, would be the answer, at least to me.
Real follow.
Sure.
How do you get that same message to your young people that you're working with?
How do you get it?
Yes.
I think, really, to me, I believe it's by doing it yourself. You know, in other words, if you don't do it, you know, I mean, to me, it's by example. And not only is it me doing it, like we were talking at lunch, I mentioned different people, like, you know, my postdocs and graduate students, like David Edwards and Ram Sasaskaran Ganesh, so that they see other graduate students and postdocs doing it, and so they see this whole group of people doing it, and so to me, role models is a very, very key thing throughout.
Who was your role model?
So for me, I didn't have a role model in that, but I did have a wonderful role model in Judah Folkman, who was my postdoctoral advisor, and I should go over that. Nick talked about it some. So when I graduated with my degree in chemical engineering, I did something that at the time was really very unusual. I was a postdoc at Boston Children's Hospital in the surgery department. And I was the only engineer there for a while. But Judah Folkman, who was a visionary surgeon and really came up with this whole idea of stopping blood vessel growth, which has led to whole new ways of treating cancer. And when he did this, it was really widely ridiculed. There were all kinds of skeptics about it for many, many years. And the wonderful thing about him with respect to that and almost everything was that, when you talk to him, he just believed in it so much. And he really believed that anything was possible. And I think for me, seeing him feeling that anything was possible, that was a great role model for me. And I think it spilled over into my doing these kinds of things.
Other questions? Yeah. You go ahead.
I'm a first-year grad student, and I eventually was becoming an entrepreneur myself. What advice would you give your other grad students and pursuing scientific research and things we should do to become entrepreneurs, things we should avoid both in the entrepreneurial world and science?
Well, I think it depends on what stage you're at, but I still feel if you're an early stage graduate student, I still think the best thing to do is do the best research, really try to do something for your thesis that's really important and do a great job on it, publish it, patent it. That may or may not lead to the thing that you want to do per se, but I still think that that's the right thing to do. I also think it's worthwhile to be exposed to different kinds of things. I don't know all of the programs they have in Texas and here, but like MIT has these programs like a 100K competition where they have business plan competitions, and students do get involved in different entrepreneurship things. And I think, I would not say spend all your time on that, but I think getting exposed to some of that is also good.
There's a question over there?
I am a professor of mathematics here. I'm a founder of a company in the Valley, doing communication. And my question is the following. So what you do in your research, of course, affects the shape the company that you found, but it also goes in the reverse direction. And the things which are done in the company, reverse back and influence your research. And then how do you mitigate the delicate issue of conflict of interest? That your students are working on things which are related to things which are done in the company.
Yeah, well, MIT, and I'm sure here too, I basically, my motto is to just follow whatever MIT's rules are. And MIT's rules are, if you have equity in a company, the company can't fund your lab and things like that. You can learn something at that company. And if you want to do research and you have some kind of funding, I'd say MIT would say that that's probably fine. But if you make a new patent in your lab, MIT will not necessarily give it to that company. They may feel that that's a conflict. So, to me, it's a decision that our technology licensing office would make. And it may very much depend on the original license that they granted to that company. But they have a set of people there that I think are extremely good, led by Lita Nelson and Lauren Foster. So they handle that. But I would just, my rules are just generally, total disclosure and whatever else the MIT would require.
Yeah, there's a question down there.
I was just wondering, kind of more environmental related, what do you think about gene patents when it comes to the conflict of both promoting and also, in some sense, scientific certain types of research?
I'm not sure I understand. Could you repeat?
Gene patents, basically. What,
Gene patents.
You have, essentially, how do you think that, essentially, I know it's controversial, but how do you think that it should be handled when it comes to there being conflicts between being able to start a company with your scientific research and also being able to have others perform research with these things?
Yeah, so let me break that down into two parts. One is gene patents, and I think it really will depend on what the gene patent is. There's laws on what you can and can't get, and sometimes those laws have shifted, or decisions, at least in the courts, have shifted. So I think a lot, but I don't know that I think the gene patents per se, other than that issue about what you can and can't patent, really relate to the second part of the way I'm interpreting your question, at least, and you can extend it if you want. But the way I look at it is a patent in general, can, whether it's gene or otherwise, can prevent people from doing things. To me, what I've generally found in the medical area, and this is a generalization, but most of the time, if you don't do it, like I said, it's not going to get done. And I find that if I really want to get a good investment to make something happen, I've got to have good intellectual property. And if I don't have intellectual property that blocks others from doing it, it's going to be harder for me to get that investment. It's going to also be harder for me to get that investment from companies. So I think if it's a legitimate patent in the gene area or other areas, I personally think it's okay, and I think that those things have largely worked out over time.
I saw some hand over there, yeah, and then, go ahead.
You mentioned in the building a company, pick a great CEO, and then move forward. What are the attributes you put on a CEO that makes you think at the onset it'll work out?
Great question for which there's not a great answer. I mean, picking great CEOs is tough. I mean, I'm not sure there are any rules, but the kinds of things that you look for are people who are very bright, not necessarily that they're an expert on the science, but extremely bright, passionate, asking really good, deep questions, being able to size things up quickly, that they have certain types of negotiating skills. For example, if the CEO came in and said, well, I'll be CEO for $50,000 a year, you probably would think that person's not going to negotiate a good deal for the company either. And so, on the other hand, if they said, well, I want $2 million a year, you'd think, well, the person's not practical. You learn a lot from, I learn a lot from the questions people ask me. Very hardworking, passionate. But a lot of times what I do is just checking what other people say about that person that's really worked with them, and to try to get a sense of how good that person really is. But a great CEO, at least in the BioWare, they have to be able to raise money, they have to be able to have a vision, and they have to be able to hire great people, and so forth.
We have a question here and a question in the back. Go ahead.
How do you get the academic world beyond the notion that publishing is sufficient?
Yeah, well, I think the way that happens, and possibly I'm an example myself, and there's other examples too, I think that the world today more and more is judging things by impact. So I think if you, and I feel, and I think this is true for me, like Nick said, I have probably 100 or more papers in Science and Nature. That's good, but the fact that those have led to products, I think when people talk about what I do, or they think about awards or honors, or me in general, I think that the fact that we have products as well as those papers, the products mean more, because they've impacted people's lives. I think that, and I'm hardly the only one. I think there are also, the media can have a huge effect. In our case, the New York Times wrote a significant article on what we do a year and a half ago, and what was striking to me when they wrote that article, it was actually the front page of the business section. One person told me that the dean of the medical school at Case Western Reserve, and as well as the dean of the medical school at UCLA, sent that article to every single faculty member there and said, why aren't we doing more of this? So the media can touch far, far more, I'm talking to, a few hundred people here, but the media can have an enormously positive or negative effect. But in that case, I think the New York Times, the woman who wrote it, she said it was the second most biggest article in terms of people looking at it for the New York Times that day, and Sunday Times, and so it was. So I think that the media can do a lot. I think universities can do a lot. I think MIT and Stanford do reward and celebrate these kinds of things. And it's not the only thing, nor should it be. But I think that but to your question, which I think is a very good question, I think places like MIT and Stanford have gained from not just the science and engineering, but from the translation. And I think that that's made their reputations so much the better for it, whether it's students wanting to go there or donations from wealthy alumni and ratings, all kinds of things.
I'll take one last question, and then I have one closing remark, and then I'll pass it to Bob Metcalfe.
Yeah, so the rules that I've used for winners, and I do think this would work. And again, people may get mad at me because they may not fit these criteria, but but I do feel like one way to judge a winner is by the quality of the publication. In other words, I feel that if you do publish something in these high-profile journals, like Science or Nature, that doesn't necessarily mean it's even scientifically, sometimes those journals you've had retractions and stuff like that. But in terms of impact, those journals are pretty good at I mean, I shouldn't say this as a chemical engineer, but I will. If I were picking a study in Science or Nature and compared that impact to, say, AICHE Journal, it would probably be, I'd be more likely, if I were in the tech transfer office, to pick the thing in Science or much more likely. So the way I think you pick winners is you can have help on those winners, because if something's been published in the really top places and gone through both the scientific and editorial review, it just to me seems like it has had a higher chance of success. Yes? Well, they come up with ideas and publish there. I mean, but
Well,
I mean that's coming up with big, and I'm sure, and some of your scientists have, you know. But the way you do it is you come up with big ideas, you raise the money internally, you know, at the University of Texas, and you shoot for breakthroughs, and you publish in those kinds of journals.
Aaron, thank you for this last question, because this is perfect really for how I'm going to close it. In June of 2013, in Nature Biotechnology, there was a very nice article by Bob, and then a postscript by somebody else, Building a Business, a personal account of translating discovery in an academic lab. If you haven't seen it, it's in volume 31, number 6, page 487, June 2013. And he gives us examples of how you pick winners and how you're successful. And 2 boxes, the story of Enzatech and the story of Momenta. And it's followed by a postscript called the Langer Lab's Secret Sauce. So this is really only the beginning, and I'm pretty sure we'll have time to continue the discussion, perhaps even over drinks. But I personally thank you for giving us some of your wisdom. I've followed you for 40 years, has been 44 years, my God, Bob. And it has been really always a pleasure to interact with you. Bob?
So let's break now and then return promptly at 3 p.m. Where Dr. Langer will give some advice to Dean Johnson on, is that okay if he gives you advice?
I'll take advice from Dean Johnson.
On how to build an innovative medical school. So please come back at 3.